Thank you very much. And in the sessions this afternoon, I've heard a lot about so what. There's a lot of amazing data, obviously available now through UK Biobank. But what does it mean in terms of what potential changes in outcomes for patients? What does it mean for services? So I'm going to just tease you a little bit about stuff that we're hoping to do with UK Biobank in terms of brain health and how that could feed through to important changes for services. So starting just with a few statistics, which I'm sure you're all very aware of. Obviously dementia, and apologies, my slides seem to have morphed a bit on the wording. But obviously dementia is one of the huge health crises that we're all facing, increasing numbers being affected in the UK, and particularly now lower middle and income countries. And these are still forecast to triple over the coming decades. The difference, speaking as an old age psychiatrist who's been working in this field for 30 years, is that we do seem to have disease-modifying treatments now, approved here and in other countries. We also know an awful lot more about risk management in terms of progression of dementia, and the last Lancet Commission paper here is highlighted on the right-hand side and demonstrating that potentially 45% of dementia cases could be modifiable if we could manage to identify and crucially help people modify those risk factors. I think the other key thing is that patient expectations are shifting. Again, if I look back at memory clinics I did 30 years ago, we were still talking to people, or people were coming to us and talking about, I think I've got senile dementia, or this is just senility. That's massively shifted now. People don't just want a dementia diagnosis, they want to know what the etiology is. And even if they're at risk or it's early disease, they want to know what type this is. And I've sat in a lot of meetings over the last few years with colleagues where there's a debate of, should we be providing that information when there isn't a treatment available at this time? And I think we're all moving away from that slightly paternalistic view. But it's partly because I think patient understanding, education is shifting. They want to know how they can modify their risks. And this is partly shifted by where we are with biomarkers. So for the first time last year, the Alzheimer's Association in the US has driven the idea that we should be diagnosing Alzheimer's disease primarily with biomarkers in mind. But the reality on the ground in clinical services is very different. We very rarely use biomarkers in terms of fluids. Only 2% of people in the UK have access to PET or CSF testing for dementia or diseases that can lead to dementia at the moment. And I would say even in terms of clinical diagnostics, we're still using ICD-10 codes that diagnose mild cognitive impairment as one group. So even in terms of how we're recording clinical data, we are way behind, I would say, a lot of other specialties in medicine, but we need to change. And I think the type of data we can derive from UK Biobank will be crucial to that. So I'm just going to tell you a little bit about what the UK Biobank Brain Health study will do. It's going to start collecting data, new data, from UK Biobank participants who have self-reported memory problems, an existing memory disorder diagnosis, or Parkinson's disease or mild cognitive impairment. And there was a lot of debate about how we would identify these people, whether we again would scrutinise primary care records, secondary care records. And again, I come back to the quality of data that's out there. So this is a starting point at least. Obviously, the opportunities are to be able to start to detect associations, particularly between risk factors and neurodegenerative diseases at various stages in such a large cohort. And the protocol as a whole was put together over the last months, really, with various working groups looking at the components and a strong input from PPIE. The underlying aim is to enable better characterization of people with neurodegenerative diseases in UK Biobank and their identification, and principally identification of subtypes. So in terms of what's happening at the moment, stage 1 has just finished, hopefully, which was an internal pilot led from the Stockport Imaging Centre for UK Biobank. And this was really about acceptability and feasibility. What impressed me about this was the group of people that were identified in UK Biobank within a 20-mile radius of this imaging centre. So actually a really large number of people. I will draw your eye to the fact, though, out of this large number, only 140 people had a diagnosis of dementia or Parkinson's disease. I don't believe that. So again, it says something about how well we're diagnosing people in clinical services, and also crucially how we are recording that. Because then if you look at people who have reported symptoms, it's a much larger number. So once we've established the feasibility of this via the stage 1, The plan is to move on to an external pilot where there will be about 100 participants that will be undergo the protocol at 2 to 3 external provider sites. And then the main phase of the study will invite about 10,000 participants at multiple UK Biobank and provider centres. Just to give a sense, these are really small numbers, but in terms of people that have come through to the initial pilot phase, what I guess is nice to see is that there's a fairly good mix of people, as you would expect, with Alzheimer's diagnoses, large number with mild cognitive impairment and other dementias, as well as Parkinson's disease. So we are seeing a good mix of people. And this is what the protocol will look like. So importantly, MRI imaging, there was a lot of debate whether we could get away with using a 1.5T scanner or whether we needed to have 3T. And I'm certainly pleased that we're going with 3T. My understanding is that this is still to be defined, but it will incorporate the existing UK Biobank MRI protocol minus the tasks, probably ASL incorporated and maybe an iron-weighted sequence as well. Blood biomarkers will be collected, and also really interestingly, some more scalable potential biomarkers such as smell tests. And again, I think this speaks to Pierce's comment about if we could find a more scalable assessment to identify people earlier, that could be a real game changer for clinical services. And then obviously a clinical assessment, so cognitive tests, questionnaires with caregivers or family members to ascertain any behavioural or psychiatric difficulties. And then a section that will include wearable devices. Again, really interesting areas of research in terms of how we identify people earlier on. And again, I'm just going to emphasise, I'm sure you're all aware why this is important. These are a variant of the Jack curves that get put up at pretty much every dementia talk that happens. But of course, what we need to be crucially doing is identifying people either in the prodromal or preclinical phase, particularly of Alzheimer's disease. And you can see on the curves here demonstrating where we are now to a certain extent with existing fluid and imaging biomarkers to identify those people. We have a lot of drugs in the pipeline now, some repurposed, some new targets, and there's certainly a sense we're going to need combination therapy in the future, but they are all moving to the left. So we need to get better at identifying people with preclinical disease, and maybe in time, I would hope, thinking about actually primary prevention and identifying people before they get the disease, mindful of the fact that we now know Alzheimer's disease probably starts in midlife, so we are going to have to go earlier and earlier. And obviously, ultimately, the vision is that in future services, we will be giving people a probability of neurodegeneration rather than a diagnosis. What is crucial, of course, is we are going to have to completely rethink what we do in clinical services at the moment. I did my first memory clinic 31 years ago now, and I still do the same thing in my memory clinic as I did 30 years ago, I'm afraid. So we've got to change. Because we've got to use biomarkers to identify any shift, any response to treatments, because we're going to see people with minimal cognitive impact in clinics, hopefully. And so obviously the ultimate goal, or the initial goal, is to think about secondary prevention, where we would work with people to look at a combination of their risk factors that could be modified, probably their cognitive profile rather than impairment, and their biomarkers, potentially to start to develop modelling for an accurate prediction score. And then importantly, we've got to tailor treatment and be able to measure response. And as I said, that's going to get more complicated the earlier we see people in the pathway. But I took this slide actually from a colleague's slide deck, and I think it's just a nice demonstration of what we could be doing in memory clinics in the future. So you would start to define people's various risk factors, whether that's lifestyle, genetics, specific biological risk factors around things like diabetes. And over subsequent years, you would target management. So in this example, you target somebody's management of their diabetes and their weight. 10 years later, the risk factor associated with the diabetes has reduced. So although their biological risk because of their age is increasing, overall you've been able to shrink their risk of dementia. And then it may be only later in life that you need to think about targeted drug therapy. Or as my colleague said, because it's the NHS, you're only getting to see people every 10 years anyway. But let's hope not. So how are we going to deliver this? There have been many papers coming out over the last 10 years, I would say, about how we need to change clinical services. And there was a publication a couple of years ago now about what needs to change in European services and what the emphasis needs to be. And you can see here on the top right, it's a strong emphasis on risk assessment, personalised prevention, which again speaks to the need to identify disease more accurately. And one key paper that came out in 2017 was something called the Edinburgh Consensus, which said what I've just said. There's a lot that we are understanding now in terms of biomarkers. Clinical assessment has changed. We've now got new therapies available, at least approved to a certain extent, and yet we are still doing the same things in clinic. So just to finish, I wanted to highlight that we are starting to see changes in clinical services. I co-lead one of something called the Brain Health Coalition network, and it's about a dozen centres that are broadly using research money to modify the clinical pathway with a real emphasis on early and accurate diagnosis, and particularly people in that prodromal group. I'm part of the Oxford Brain Health Clinic, which is led by Claire Mackay and Lola Martos, which is looking at can we actually implement these assessments in the real world. And I'm pleased to say we've now done this in almost 500 people. This just emphasises that the assessments that people go through at that clinic now incorporates traditional clinical assessments and research assessments. And the nice thing for the commissioners and hospital managers is that straight away we were able to reduce their consultant appointment from 75 to 45 minutes, so immediate cost saving, which is always what people look for. And in terms of acceptability, we have a really high level of consent to come and do these extra assessments. You can see highlighted on the bottom left. And in terms of feasibility, the top right, you can see that the mean age about 2 years into our pilot was 78 and our oldest participant was 101. So these assessments are acceptable and feasible to do in a large number of people. And I'm aware I'm probably running out of time, so just to finish with a little bit of science, which I will credit to Ludo Graffanti and others in our group, and Ludo's here still, I think. Again, how does the UK Biobank data help this process? Well, the imaging protocol in the UK, in the Oxford Brain Health Clinic, has been aligned with the UK Biobank MRI protocol. And what this is enabling us to do is to use those norms, but also identify some gaps in the norms that we have, particularly in the very elderly, the over 80s, but also potentially gives us the data to identify people earlier via this type of imaging protocol. So this graph nicely demonstrates by the red dots individuals' hippocampal volumes and how they are obviously outliers from what we would expect based on UK Biobank data. It's also already showing promise in terms of increased confidence of diagnosis, and Ludo and Claire are using this to start developing a pipeline for automated extraction of brain health information from the imaging PACS data so that we can have improved reporting for clinicians at the end of the day. So in terms of what we're doing with data that we have already and what I would hope we could do with the UK Biobank brain health study is think about how biomarkers, whether it's imaging, whether it's fluid, whether it's wearable devices, can be used to give better diagnoses, more accurate diagnoses, and use that data to help improve services for everyone. Thank you.